Assalamu alaikum wa rahmatullahi wa barakatuh. My name is Ayham Nabhan, a third year medical student, and today, inshallah, I'm going to be your tutor for amino acid and titration curve lectures. So, as we used to before we start each lecture, we need to read this dua. اللهم إني أسألك فهم النبيين وحفظ المرسلين والملائكة المقربين. اللهم اجعل ألسنتنا عامرة بذكرك وقلوبنا بخشيتك وسري بطاعتك إنك على كل شيء قدير وحسبنا الله ونعم الوكيل. Let's get started with our lectures with the amino acids lecture. So let's dive into the structure of each amino acid that is made in our body. But before we talk about the chemical structure of the amino acid, we need to know that the amino acids or the alpha amino acids, the dominant type of amino acids that is found in our body, they are the building blocks of the proteins that we eat, ingest, okay, or the proteins that are made in our body, such as albumin, fibrinogen, and so on, okay? Now, let's go into the chemical structure of each amino acid. So, the first thing you need to know about the chemical structure, you need to know that it is made of a central carbon, which is this one, okay? And this central carbon is going to be bonded to four structures. The first one and the easiest one to remember is the hydrogen atom. The second one is the carboxyl group, which is positively charged. Then the second group is the amino group, which is negatively charged. And the last one and the unique structure that makes each amino acid specific and different from the other one is the R side chain, which is different chemicals, okay, that makes the amino acid different from the others. Okay, we're gonna see it inshallah. So as we said, it's here, it's written over here. So the carboxyl group, okay, might get deprotonated, okay, and the amino group, it might get protonated. What does that mean? The carboxyl group, which is COOH, once it gets deprotonated, this hydrogen atom is gonna get out from the amino acid. Whereas in the NH2, okay, it might get protonated. That means we're gonna add a hydrogen atom. As you can see, it's gonna convert from NH2 into NH3, and that happens at the physiologic pH just to keep the amino acid neutral for our body, so it doesn't make any diseases and it doesn't accumulate, making any abnormalities in our body. Okay, that's it. The R group that we talked about, which is the unique structure of each amino acid, also plays a very important role in the protein folding. What does that mean? It means that the R group, okay, it might contain and make hydrogen bonds, disulfide bonds, ionic bonds, and hydrophobic bonds or interactions. Once these interactions, okay, and characteristics of the R group found in each amino acid, that would help the proteins get a very stabilized structure, making them very hard to be broken down by any pathogen. Okay, so you just need to know the basic information about the structure of each amino acid. And you can see the picture over here. Okay. So the structure of amino acid is called Zwitterion. Okay, the Zwitterion is the dominant species of each amino acid in the aqueous solutions at the pH of 7, which is the physiological pH. What does Zwitterion mean, Aslan? Okay, it means that we do have a positive charged structure and a negative charge structure of the amino acid. Those would cancel each other, making the amino acid neutral. So as it's written here, the amino acid have both a positive and a negative charge, makes it neutral. Okay, sorry guys, I said in the previous lecture that carboxyl is the positive and the amine is negative. It's the opposite actually. Okay, so once we have a positive and a negative charge, they cancel each other, making the amino acid as a neutral amino acid. Okay, that's the Zwitterion. What about the chirality? The chirality of the amino acid, okay, is going to be dependable on the groups that is bound to the carbon or the central carbon. And as we do have four different groups, the carboxylamine, hydrogen, and the R group, that would make the amino acid as a chiral amino acid or asymmetrical, because these groups are different from each other. Okay. And in nature, in the mammalians, okay, all of the amino acids are found in the L isomer. What does L mean? L means it's basically left, okay? What does left mean? Left means that the structure of the amino acid having the amino group, okay, is going to be on the left of the 3D mirror of this amino acid. Whereas just for glycine, which is the only amino acid that is found in the D isomer or R isomer, where the amino group found on the right side of the structure of the amino acid. As you can see over here, the L amino acid, the amine group, it's going to be found on the left, and the D or right is the amine group is going to be found on the right of the amino acid structure. Okay, now let's talk about each single amino acid in details. So this is like a collective table or a collective vector of each amino acid, okay, and the categories that each amino acid belong to. You can just memorize it by your own once we're done with the details. So let's start with the first cyclic secondary amino acid, which is proline. So why are we saying it's cyclic? Basically, proline is a cyclic amino acid because the structure between the amine, okay, and the R group makes a cycle, okay, where the chain, okay, like it folds and gets back into the R group. That would make it as a secondary amino acid. Where in the primary amino acid, as you can see here in the alanine, there is no cycling, there is no folding, and there is no returning or coming back into the R group. Okay, so the proline is a secondary amino acid. This is a very important information. Okay, once this secondary amino acid makes its cycle or its circle, it makes something called kink. This kink is highly found or in high amounts in collagen, which is very important for the connective tissue. So the kinks, which is made from the proline, would help the collagen to give the stability and the strong structure of the proteins. That's why our connective tissues, okay, are very strong because they have this proline amino acid having this stabilization of each amino acid or of each protein, sorry. Okay, okay, okay. Let's go now into the other group of amino acids, which is the branched amino acids. What does branched mean? Branch means it has many divisions, okay, along the axis. So as you can see here, we do have three branched amino acids, the leucine, isoleucine, and valine. You can see along the axis, we do have a branch. This is the first one, second one, third one, fourth one. In isoleucine, we do have 1, 2, 3, 4, 5. In valine, 1, 2, 3, 4, 5 as well. Okay, so it's a very and highly branched amino acid. It's due to the branched chemical structure itself. Okay, okay. So there is an important feature and characteristic of these amino acid. What is it? It is actually these amino acids, okay, need an enzyme or needs enzymes, okay, to be metabolized or to be broken down. حلو? حلو. So once these enzymes are deficient or they are not available, okay, they won't be broken down, thus they would accumulate, right? Okay, give me an example, Ayham. An example of this accumulation is the maple syrup urine disease, where the enzyme, okay, for the branched amino acids to be broken down is deficient. So they would accumulate, okay, and the urine is going to be maple or is going to be maple syrup taste, okay. So that's for the branched amino acids. For the sulfur-containing amino acids, where they would have a sulfur or sulfur structure, okay, they're going to be two amino acids. The first one is going to be the methionine. حلو. The second one is going to be the cysteine. Methionine, let's talk about methionine first. Methionine is the first amino acid to be brought once we're doing a protein, okay, with the AUG, which is the starting codon. So during protein synthesis, we're going to start the chain of a protein or the peptide chain. The first amino acid to be added to this chain is going to be methionine, okay, along with AUG, which is the starting codon to start the protein synthesis. Okay, okay, nice. Also, methionine, okay, you're going to take it later on. Methionine is a precursor of something called S-adenosyl methionine, or SAM. You just need to memorize the name. You don't need to know the details about it, because if they ask you about it in the midterm, they're going to ask you what's the precursor, or methionine is a precursor of which molecule. You're just going to answer S-adenosyl methionine, okay? Okay. This is the second point about metabolism, methionine, sorry. The third point that you need to know, and it's a very important point, you need to know that methionine is essential and important for the methylation process or reaction of DNA, proteins, neurotransmitters, and lipids. This methylation reaction or this methylation function of methionine makes all of the following structures active, okay, and functional. Cool? Okay, that's it. For cysteine, which is the second sulfur-containing amino acid, it has a very important characteristic, okay, which is just found in cysteine, which is the sulfhydryl group, or SH, okay? This sulfhydryl group serves as a site to make a disulfide bond between two cysteine molecules, okay? So as you can see here, this is a cysteine, this is another cysteine. Once they, or once the sulfur ends bind together or meet and bind together, they make a disulfide bond. What does disulfide bond mean? Di, two, sulfide, two sulfur molecules connected by a bond, okay? So this disulfide bond, from its name, disulfide bond, and it's the bond, and the bond is just for stabilization and strengthening of the structure. So it's basically just to stabilize the protein's tertiary structure, specifically in albumin and fibrinogen, okay? So if a question in the exam comes to you and saying, which of the following proteins are stable contains a high amount of cysteine having a disulfide bond to stabilize its structure? What would you choose? You're going to choose albumin and fibrinogen, or fibrinogen, depending on the question. Okay, so that's the sulfur amino acids. Next, we made the amino acid, okay? What do we have as a post-translational, okay, modification of an amino acid? Okay, we do have two modifications actually. We do have the phosphorylation and we do have the glycosylation. حلو, حلو. Let's start with the phosphorylation. Just to know that phosphorylation and dephosphorylation is very, very important for the enzyme's activity that is needed for our human body functions. Okay, okay. So the phosphorylation would happen for serine, threonine, and tyrosine amino acids, where they have hydroxyl group, which is an attachment site for the phosphate group. Okay, so these amino acids having hydroxyl group, which is an attachment site for the phosphate, once it binds to it, it might phosphorylate it or dephosphorylate it depending on the enzyme that is working. صح ولا لا؟ صحيح. So if we have an enzyme ending with a kinase, okay, like phosphofructokinase مثلا, the phosphofructokinase or the kinase, it's going to add a phosphate group, okay, to the amino acids, making them functional, making the protein functional, making our body functional. So that's kinases. What about phosphatases? Phosphatases, from the name ases, it removes the phosphate group from the amino acid, makes it unfunctional, okay, or dysfunctional, or whatever, okay. So phosphatases removes the phosphate group. What about glycosylation? Glycosylation, as the name indicates, it's glucose, fructose, sugars, صح? So it's basically, okay, a reaction where they're going to be an attachment site to add a sugar group such as oligosaccharide, okay, in the glycoproteins, okay, to make it as a bigger glycoprotein, to break it down, to make it bigger, or whatever process needed for the body. Okay, okay. What are the examples of glycosylation modification? We do have three amino acids: asparagine, which has an amide group, okay, and serine threonine, which has a hydroxyl group. The amide group and the hydroxyl group serve as the sites for the glycoprotein or for the oligosaccharide to be added to this amino acid, making the glycoprotein. Clear? As simple as it is. Okay, so those are the modifications of the amino acids. Now, once we want to talk about the polarity of the amino acids, we do have polar, non-polar amino acids, acidic, basic amino acids, and so on. Let's start with the polarity. We do have polar, which is hydrophilic, non-polar, which is hydrophobic. حلو حلو. During the protein folding, okay, and the synthesis of proteins and amino acids, we do have hydrophobic, okay, or the non-polar R groups or amino acids, okay, having the non-polar R group, okay, those non-polar would be poured inside the plasma membrane. So as you can see over here, this is an amino acid. Sorry, this is a protein, okay, having its own amino acids. The non-polar or hydrophobic amino acids are going to be poured inside or along throughout the plasma membrane, as you can see over here. Whereas the amino acids that are, that is making the amino acids that are making the protein, and they are hydrophilic or polar, they're going to be facing the extracellular and intracellular fluids or the يعني external surface of the plasma membrane. Okay, so that's it. So as you can see over here, in membrane proteins, hydrophobic amino acids face the lipid membrane. As we said over here, they're going to be throughout the plasma membrane. Whereas the hydrophilic amino acids, they're going to face the extracellular or intracellular spaces. And that's it. Okay, so that's for the polarity. Now, the first category, which is the non-polar amino acids. The non-polar amino acids are 9 amino acids that you need to memorize them, as you can see over here. The first one is alanine, then valine, methionine, leucine, isoleucine, proline, tryptophan, phenylalanine, and glycine. But you might ask me, why glycine isn't in the graph or isn't between these non-polar amino acids? Because I'm going to tell you why. Because it's a tricky amino acid. Why it's tricky? Because it actually might pretend as a non-polar amino acid, or it might be polar amino acid, depending on the environment where glycine is in. Okay, okay. So for the non-polar amino acids, why are they not they are not polar? Because they don't have any R group, okay, having any chemical structure that makes them polar. What are the polar R groups? Okay, they are hydroxylaminamide and the sulfhydroxyl. Okay, so these R groups with these chemical structures would make these amino acids polar but uncharged, because there is no reaction that is making them charged, okay, or uncharged, whatever. So the amino acids, the polar amino acids, are tyrosine, threonine, glutamine, serine, cysteine, and asparagine. And as I said, glycine, which might act as a polar amino acid. Okay, you need to memorize them, and you need to memorize the chemical structures over here, which is the or chemical groups, which is hydroxylamidamine and sulfhydroxyl. Now let's go into the two most important amino acid groups, which is acidic and basic. The acidic amino acids, they are just two, glutamic acid and aspartic acid. And as they are acids, so they donate with a proton. They give a proton for the basic amino acids. We're going to talk about this later in the titration curve lecture, okay? So at the physiologic pH, the pH of 7, okay, the side chains, they're going to be fully ionized, okay, with a negative carboxylate group, okay, and a positive amine group. As we said, the fully ionized amino acid, it's going to be called aspartate and glutamate, where they're going to be neutral, okay? They're not going to be negative carboxylate. حلو حلو. Or they're not going to contain a negative carboxyl group. They're going to contain a neutral carboxyl group, which is going to bind with a proton, or with a proton, which is H, or hydrogen atom. For the basic amino acids, there are 3: lysine, arginine, and histidine. Okay, those basic amino acids, they actually accept protons from the acidic amino acids, and at the physiologic pH, they are positively charged, as they are basic or alkaline. Now, one of the most important clinical ideas or clinical things that the doctor talked about, which is the slow, long-acting insulin, which is made in lab to be given for diabetic patients, okay, just to make and maintain a longer action, okay, making them easier to use. حلو حلو. So the modification that makes these structures available for this insulin, okay, or make these features available for this insulin, is an easy modification, okay, where asparagine, okay, is going to be changed with glycine at the position of 21 in the peptide chain for insulin, okay. Okay, what is the effect of this change? The effect is going to be a less soluble structure or insulin in water, which is going to make it absorbed slowly from injection site, okay, and that would provide a longer stable action of insulin, okay, where that would help to maintain a stable blood sugar in diabetic patients. حلو حلو. So it's basically just a clinical use, okay, and a clinical modification of insulin to help diabetic patients to have lower injections of insulin, making them very long acting injections. حلو. Dive into the next part of our lecture, which is going to be the titration curve. But before we go into the titration curve, we need to know what is the pH scale. Okay, the pH scale and the acid-base balance. Okay, we talk about that pH measures the concentration of hydrogen in our body. And the human body would maintain a pH between 735 to 745, which is the hemostasis of our body, keeping our body and our functional reactions and our functional processes good and under the maintenance of this pH. حلو حلو. The second point that you need to know is that the biochemical reactions in the cells ICF, okay, would produce acids that must be balanced with bases. So the overall cell nature and environment is to be neutral, okay? Okay. Just know any reaction or any process of our body need to be neutral. حلو حلو. طيب, this is the pH scale as you can see over here on the right of the slide. Okay. This is the baseline, or this is the neutral pH, which is going to be 7, as we said. Once we lose hydrogen ions or we take hydroxide ions, okay, the pH is going to increase, be more alkaline, be more basic, and the number would increase طبعا, as you can see over here. Once we go down less than 7 pH, okay, more hydrogen ions would come into the amino acid, making the amino acid more acidic, as you can see over here. And the number is going to decrease as well. So it's written over here, a pH of 1 is going to indicate a very strong acid, which release hydrogen ions, okay, to neutralize the environment. You need to know this point. A pH of 7, which is neutral, okay, the concentration of hydrogen and hydroxide, okay, is going to be the same concentration, or is going to be equal. حلو حلو. A pH of 14, which is the most alkaline, is going to be a very, very, very strong base where it releases hydroxyl ions. حلو حلو. pH more than 7 is basic, and a pH less than 7 is acidic. That's the basic information that you all need to know in this slide. طيب, summarizing Dr. Abdul Jabbar lecture in one table, okay, you would have this table. So, what is the difference and relationship between pH and pKa? So, let's break it down into small details or into small points. حلو حلو. So, the first thing, what is pH? pH is the log or the logarithm of the concentration of hydrogen or the protons in our blood, in our body. Once we take the log, the pH is gonna appear. حلو حلو. So, it actually measures the hydrogen ion concentration in a solution or in our body, as I said. حلو حلو. So the lower the pH, the higher the concentration of hydrogen, the more acidic the solution is. صح؟ صح. So if we have two solutions and they are both acidic, but the first solution is very concentrated with hydrogen ions, okay, the other one is diluted. حلو. Which one would have a higher pH and which one is going to be stronger and more acidic? The first or second one? The first one, because pH depends on the concentration of the hydrogen. So once it's a very concentrated weak acid, it would have a lower pH, more acidity. Okay? Okay. That's pH. The second point or concept is the Ka. Okay, what is Ka? It is basically the acid dissociation constant. So it's a constant value of each amino acid. حلو حلو. So it measures how a weak acid dissociates into a hydrogen and its conjugated base. We're gonna talk about this later on. So the higher the Ka, the stronger the acid, and the property of the acid is going to be independent of the solution concentration. مو مثل pH, not like pH. pH is going to be dependable on the concentration of hydrogen in a solution. Ka is not dependent on the concentration of hydrogen. Going to the third concept, which is pKa, which is going to be the same, which is going to be the logarithm of Ka, or the value of Ka. It actually indicates the pH at which a weak acid or base donates or accepts a proton. So basically, the lower the pKa, the stronger the acid, and that would define that the effective range of a buffer is very efficient and very effective for our body, making the body balanced and neutral. حلو حلو. The acid and base. The acid, as we said, it donates protons. The base, it takes protons. That's it. Okay? And the key information about the acid and base, they actually react together and make reaction and processes to make buffering systems to neutralize our body and our body compartments. حلو حلو. pKa and pH has a similar equation called Henderson-Hasselbalch equation. This equation, it actually shows the relationship between pH and pKa and the ratio between the weak acid and its conjugate base. حلو حلو. And it's very, very useful for calculating the buffer pH and to understand the values of each of the pH and the pKa. We're gonna talk about it in details. But before we go to the next slide, you need to know that each change of one pH unit values 10x difference in the concentration of hydrogen. So, as an example, a pH of 2 has 10x more hydrogen than pH of 3, and so on. Okay? Okay. Now, as I said, we're gonna talk about Henderson-Hasselbalch equation. And as we said, it just shows the relation between pH and pKa. حلو. And the ratio between weak acids and its conjugate base. So let's break it down into an equation. This is the equation. This is pH. Okay, or rather, this is the equation. We're going to start with the value that we want to get, with the thing that we want to get from the compartment and from the structure, which is the pH. حلو. Which is the logarithm of hydrogen. Lower is more acidic, and so on. The details we explained. So we want pH. How do we calculate pH? We calculate it by having pKa, which is going to be a constant, okay, for each acid, plus the log of concentration of the conjugated base over the concentration of the weak acid. حلو حلو. You need to know this equation, and you need to know the breakdown of this equation because it's very important equation. They might bring it in the exam. They're going to give you three values and ask you for one of the definitions. Might be pH, pK, A, or HA, which is the base or the acid. So it's over here written. No, I need a breakdown of this equation is written over here. طيب, let me explain mathematics now. If we have a solution, okay, with a very high amount of the conjugate base, would the pH be lower or higher than the pKa? It's a direct proportion relationship between pH and the concentration of the base, means as we increase the concentration of the conjugate base, the pH would increase, meaning it's going to be higher than the pKa. And as you can see over here, once the base is more than the acid, then the pH is going to be higher than the pKa. And that would make the solution basic because we do have a higher concentration of the base. Common sense. طيب, what if we do have higher concentration of the acid, which is in an inversely proportional to the pH value? So once we increase the acid, the pH would decrease. So if the base is less than the acid, means that the acid is more than the base, okay? The pH would be less than the pKa, meaning the solution is an acidic solution because we do have a higher concentration of the acid more than the base. حلو حلو. That's it. Easy equation. I hope you don't go into the details of it. You just need to memorize this equation and memorize these two definitions. That's it. Now let's go to the bulk of this lecture, which is going to be the titration curve itself. So the titration curve I would break it down into three parts: the buffer zone or the flattened zone, the acidic zone, this one, or the acidic part, and the basic part. Okay, okay. So this point is going to be the isoelectric point of an amino acid, where the amino acid is neutral, there is no charge, okay, and there is no change of charges, there is no loss of protons, getting low protons or whatever, okay, okay. So this is the buffering region. It might be one or minus plus or minus 1 pH unit of pKa, okay. But once we go to the right, meaning we are increasing the concentration of A, which is the base, that would make the titration curve more basic. That would make the amino acid more basic. So once we go to the right, the base is gonna be added, making the amino acid basic or alkaline. Once we go to the left, okay, that means we are donating protons, we are losing protons, okay, and adding acidic solutions or acidic molecules, which is the HA represented in the equation, okay, the titration curve, okay, would decrease and the pH would decrease and the pKa would decrease. That means that the solution is being more acidic and a stronger acidic solution. That's it. As easy as it is. We go to the right, it's more basic, pH and pKa increase. We go to the left, it's more acidic, pKa and pH decrease. Once we go to the right, meaning we add bases. We go to the left, we are adding acids, okay? And we have in the middle, which is the flattened region or the buffering region, where the pH and pKa is gonna be just plus or minus 1 difference. That's it. Okay, okay. As an example of titration curve of a certain of a certain acid, it's going to be the acetic acid. Okay, you can see that the buffering zone or the buffering region is going to be around 4.76 pH pKa. And once we increase the base, okay, once we increase the base, meaning we are losing hydroxyl group, it's going to be more and higher pH. Once we decrease it, okay, it's going to be less pH, okay, adding a proton as well because it's an acidic solution, acidic part of the amino of the solution. Okay, okay. That's the first 2 points that we needed to explain. The third point that I need to explain over here is in the buffering zone. Here's the buffering zone, okay? Oh, sorry. Here's the buffering zone, okay? In the buffering zone, the pH and the pKa is going to be the same. Hello? They're going to be equal to each other. Once we go to the right, making the solution more basic, pKa is going to be higher, pH is going to be higher than pKa. Once we go to the left, we go down, making the solution more acidic, the pKa is going to be higher than the pH. طيب, you might ask me, where did we take it? It's over here, as you can see. Basic, pH is going to be more than pKa. Acidic, pH is going to be less than pKa, or pKa is greater than the pH. That's the third point we need to explain in the titration curve. Titration curve done. Inshallah, you're going to ace it in the midterm and the final. Now, let's talk about the buffers and the buffering systems, which is the last point, I think, or the last part of the titration curve lecture. So, we do have many buffering systems, many buffering chemicals that neutralizes our body, maintaining the hemostasis of our body just to keep every single process functional, okay? And we don't lose any part of our intracellular components. What does a buffer do? It actually help to minimize the pH change when an acid oral base is added to a neutral solution. Okay, so they would contain these buffers. They are made of a weak acid, okay, HA, and its conjugate base, which is the A. حلو حلو. The acid, okay, neutralizes the added hydroxyl group, whereas the base neutralizes the added hydrogen atom or the proton. Why? Because uh, plus or any if we have an equation having a plus and a negative or a positive and negative charges, they're gonna cancel each out. صح ولا لا? So the base, okay, is gonna cancel out the acid, and here the acid is gonna cancel out the addition of a base. حلو حلو. So the idea of this buffer just to keep pH stable and near the pKa value, تمام, which is going to be, as I said, plus or minus 1 of the pH unit. تمام تمام. So at the pKa, the concentration of the weak acid and its conjugate base is going to be equal, because that area, the buffer would work the best over there. You might ask me, where can we see it in the titration curve? It's this point, which is the buffering region. The buffering region where the pKa is going to be equal to pH, and the buffer is going to be working at it at its best. تمام? تمام. So let me explain this equation. So we do have a buffer, okay, having a weak acid and its conjugate base, okay, and we do have a solution over here. We added to the solution a hydrogen atom or a proton. Which part of the buffer would move and act on this acidic atom, okay, to remove it and minimize the change in pH and neutralize the solution? It's going to be the base, صح ولا لا? Which is the conjugate base. So it's going to be added, okay, or it's gonna act on the hydrogen atom, make a component or make a compound having this hydrogen atom to neutralize the environment. طيب, what about adding a hydroxyl group, which is a basic structure? The weak acid would move and bind to it, okay, making water. Water would neutralize the environment and the compartment, making and taking this environment back to its normal condition and state. So as you can see over here, we added a hydroxyl group, okay, so the weak acid would come and work on it. Hydrogen with a hydrogen with an oxygen atom would make water, okay, and the conjugate base is just going to be throughout, because عادي, it's fine. Okay, so that's the buffers. Let's explain the titration curve of a buffer. Okay, so all amino acids have ionizable groups, the acidic one and the basic one. The acidic is going to be the carboxyl and the basic is going to be the amine. The amino acids with two ionizable groups would have two pKa values, and the amino acids with three ionizable groups would have three pKa values. Each pKa value would represent a buffering zone where the pH is going to be resistant to change. It's not going to change. It's not going to make the solution acidic nor basic. حلو حلو. So as you can see here, the glycine, the titration curve of glycine, which has two pKa values, okay, the first one is going to be here with a value of 2.34, and the second one with a value of 9.60. حلو حلو. Each one is going to act as a buffering zone. حلو. That's it. Whereas this middle point or the isoelectric point, the amino acid wouldn't be a buffer, wouldn't act as a buffer. It's going to be a zero acting dormant amino acid, doesn't allow or act to any change of the environment. حلو حلو. As we said, we do have two ionizable groups of glycine. We do have the carboxyl group and the amino group. The carboxyl group, okay, is gonna act as a buffer for the acids. So once we protonate the amino acid with a proton, it's gonna be added to this carboxyl group just to make it neutralized. Or if we take out a proton, the amino group would act as a buffer against the added bases. حلو؟ حلو. So as you can see over here in the graph, this is the Zwitterion. It's the neutral amino acid. Once it gets protonated, we add an acid. The carboxyl would have it and acts against it, make it as a whole compound to neutralize it. Once we deprotonate, add a base or take an acid, the amino group would act on it and make it as a compound to neutralize the condition. So how is that even possible? You can see it in the equation, in the following equations. We said that the carboxyl is going to be against added acids. صح؟ صح. So once we add an acid, which is a proton, it's going to be added to the carboxyl, making a compound. Once we add a base, it's gonna be added to the amine group, making water, okay, just to neutralize the condition. So what you need to know from these equations, once we add a proton or the acidic atom, it's going to be neutralized by the carboxyl group. Once we add a base, it's going to be neutralized to water by the amine group. That's it. That what you all need to know, okay? In the carboxyl situation in this equation, the pH is going to be lower than the isoelectric point where the molecule would be positively charged because we are making a new compound. Here, the pH is going to be higher than the isoelectric point, so the molecule would be negatively charged, so it's going to be more acidic. Here, it's going to be more basic. Hello? That's it. Perfect. Last few slides. I know this slide might take a look very stressful and very crowded, but actually it's a summary of 5 slides from the lecture, and it's very sufficient and very enough for the final for the midterm. So the buffering at the physiological pH, most of the extracellular proteins that are found in the ECF, like albumins and globulins, they are going to be negatively charged. ليش? Why? Because they allow to bind hydrogen ions and act as buffers, as we explained earlier. Okay, that's what you need to know, that the extracellular proteins are negatively charged to act as buffers. That's it. Okay, okay. That's like 2 slides. Okay, the next slide is going to be the clinically modified insulin that we talked about in the amino acid lecture, but we just have a small modification or a small change. So this insulin is going to be made by replacing asparagine with a glycine at the position of 21 in the peptide chain. حلو. That would make the insulin less water soluble and has a net charge of +2, which slows its absorption. You don't need to know the value, just know it's less water soluble. That would slow its absorption. The isoelectric point of this insulin would shift from 5.4 to 6.7. So it increased, so the pKa is higher, making it more soluble at the acidic pH of the subcutaneous tissue. So that meaning we're going to get a result of a slower reabsorption, a longer duration of action, and we're going to require fewer injections with better blood sugar control. Okay? Okay, that's what you all need to know. Perfect. The second or the next 3 slides are going to be summarized in these 4 lines. Physiologically, in our body, we do have carbonic acid. Carbonic acid, this is the structure of carbonic acid, which is a volatile acid in equilibrium with carbon dioxide, which is exhaled by the lungs. Meaning, once we exhale and we produce carbon dioxide, this carbon dioxide would be reacting with the carbonic acid to neutralize the area and the compartment of our lungs and the blood that supports the lungs. So the carbonic acid is regulated by the respiratory activity. The non-carbonic acids or the fixed acids, they are not volatile and they are buffered anywhere in our body. Then once they complete the circulation, they reach the kidneys. Once they are in the kidneys, they're going to be excreted. That's it. Nothing to know about them. طيب, what about the metabolic protein that is made in our body? Okay, it actually is actually going to be buffered by the bicarbonate, which is the broken down component of the carbonic acid. Okay, so once we have a metabolic hydrogen, bicarbonate would come and bind to it. So this structure would be converted to this structure, which is carbonic acid. Then in our normal processes of our body, in our physiological processes and functional processes, the carbonic acid would be broken down into two compounds, water and carbon dioxide, to make and to keep our body safe, okay, and and take out any cause to let any pathogen come and invade our body. That's it, because acidic environment is favorable for bacteria and so on. Now, for the idea of buffering, okay, we do have three energy sources in our body. We do have glucose, fatty acids, or triglycerides and so on, and proteins. Let's start by each one. Once we do have an incomplete oxidation and non-volatile acids, specifically when the oxygen is low, such as during intense exercise, once you go to the gym, you are on the treadmill, running, 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 running. You need more oxygen to supply your muscles and your cells. So, once you need more oxygen, that means that you have lower oxygen concentration in your body. And in a shock, any type of shock, septic shock, hemorrhagic shock, or whatever, okay, the glucose is going to be incompletely oxidized because we don't have enough oxygen to be oxidized. That would make glucose be oxidized to lactic acid. Lactic acid would form lactate and a proton or a hydrogen atom. The accumulation of lactic acid and the production of hydrogen atoms would increase the acidity of the solutions of our body, okay, lowering the blood pH. That's it. That's the first point. Second, in uncontrolled diabetes, in starvation and alcoholism, okay, the fatty acid oxidation here is going to be incomplete. حلو حلو. That being said, the fatty acids would be oxidized to ketone bodies such as acetoacetic and the beta hydroxybutyric acids. You don't need to know them. Okay, these ketone bodies would dissociate into anions and hydrogen ions as well. So once we increase the hydrogen ions, we increase the acidity, we lower the pH, leading to acidosis. And it's actually known as a condition in our body called diabetic ketoacidosis because of the accumulation of ketone bodies that would be broken down into hydrogen atoms. The last part, which is the oxidation of proteins and amino acids, would produce by nature strong acids, as we said earlier. So these three systems, okay, are the sources of the hydrogen in our body. Once any one of them is defective, that will lead to the accumulation of hydrogen, leading to acidosis. Perfect? That's it. We do have three buffering systems in our body. Buffering systems might be intracellular or extracellular, just to keep a pKa and a pH of 7. The intracellular buffers, okay, with a pH of 6.9 to 7.4, they are two systems. You just need to know the names, by the way. The phosphate system and the protein histidine system. Hello, hello. I think this is phosphate and this is phosphoric acid. I don't know. I'm not sure صراحة, but you just need to know that there is a phosphate system in the intracellular compartment act as a buffer along with the protein histidine system. For the extracellular buffer, okay, extracellularly, we do have bicarbonate carbonic acid system to maintain the blood pH with a value of 7. Bicarbonate is known and it's going to be converted to carbonic acid, producing water and carbon dioxide. You just need to know the names of these systems and where are they located. You don't need to know anything else. Okay. Now to the last part of our lecture. This is the last slide. I'm going to talk in this slide about the relationship between respiration and the acid base balance. So we do have two conditions. You're gonna take it in Balmo, inshallah, like I think next week in CVB. We do have hypoventilation and hyperventilation. Hypoventilation, where the lungs would fail to exhale carbon dioxide fast enough. Okay, so that would lead to an accumulation and a buildup of carbon dioxide inside our blood. Okay, this accumulation of carbon dioxide would lead to a more conversion into carbonic acid, as we said earlier. This carbonic acid, sorry, would dissociate into hydrogen and bicarbonate. It's like the reverse of the equation that we took. We said earlier that hydrogen with bicarbonate making carbonic acid to be broken down into carbon dioxide and water. Now we're saying we do have an accumulation and a high amount of carbon dioxide that would be converted to carbonic acid. Carbonic acid would be broken down to hydrogen and bicarbonate. حلو حلو. That will lead to respiratory acidosis because we have higher amounts of hydrogen and the protons. For hyperventilation, where they're going to be a rapid breathing, that would remove carbon dioxide too quickly out of our blood and out of our lungs and body, okay, in a way that would be a very decreased carbon dioxide, less bicarbonate, less break down into hydrogen, that will lead to respiratory alkalosis, which is a higher pH. You need to know the details of each process over here because you might be asked on it because it's a very, very important clinical case over here. And I would like to end up my two lectures by saying good luck on your MOL midterm, inshallah. I wish you the best of luck, and I wish all of you, inshallah, A plus, sorry. And yeah, thank you so much. I'm sorry if that took so long because actually the lectures are so heavy. And yeah, best of luck. Please don't forget to scan the QR code. لا تنسونا من دعواتكم. تصبحون على خير. باي باي.